Today's video topic is a little different. It's on Crispr, a gene editing tool. It includes everything from CRISPR's future use to what it actually is. The first question is, what is CRISPR? CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR was originally part of a bacterial defense system, but is now used to quickly edit genes in a host organism's genome. Usually, CRISPR refers to the CRISPR-Cas9 system, which can be used to target specific areas of the genetic code and edit the DNA at these sequences. CRISPR can be used to treat the causes of many diseases. The second question is, how does CRISPR work? There are two key molecules in the CRISPR system. One is the enzyme Cas9. Cas9 is a molecular scissors that can cut the two DNA strands at a specific location in the genome to remove or add pieces of DNA into the genome. The second key molecule is a piece of RNA, otherwise known as guide RNA. This RNA is also called the CRISPR spacer sequences, and it is a pre-designed RNA sequence that is around 20 base pairs within a larger RNA scaffold. This scaffold binds to the DNA as a marker for the Cas9 for where to cut, and this ensures that the Cas9 cuts at the right part of the organism's genome. This guide RNA has RNA bases that are complementary to those of the target DNA sequence in the genome, and therefore is able to cut at a specific sequence. So after the DNA is cut out, the cell recognizes that the DNA is damaged and tries to repair it. At this point, scientists can introduce new genes into the genome of the cell. The third question is, where did scientists find CRISPR in nature? A similar system is found in bacteria that is used to respond to pathogens and viruses. The bacteria use this system to snip out parts of pathogenic DNA and store it in their bacterial DNA to recognize the pathogen the next time it attacks. This is analogous to keeping wanted posters of the pathogen in its bacterial DNA. Use is similar to a primitive bacterial immune system. The fourth question is, what can we do with CRISPR? CRISPR can be used to target and replace genes in organisms such as monkeys, rabbits, mice, pigs, and many more. In the future, it can be applied to humans to replace mutated genes such as the genes that cause diabetes and other diseases. CRISPR can also be used to induce large deletions or genomic rearrangements, such as chromosomal translocations or inversions. Other applications include transcriptional regulation and epigenetic modification at specific gene loci. For a future outlook, CRISPR holds a large potential as a tool to treat a range of medical conditions that are based in genetics, including some types of cancer, hepatitis B, or high cholesterol. Another possibility includes the editing of genomes of germline or reproductive cells in humans. However, this may not be applied anytime soon, since there are many ethical considerations since changes to genomes in germline or reproductive cells are heritable or passed down through generations. The final question is, why is CRISPR so revolutionary? There are many other types of genome editing tools that biologists can use. However, CRISPR is revolutionary since it can be used to target any gene in a target organism's genome. The only change that is required is in the crRNA or guide RNA to change the targeted gene. Therefore, CRISPR is really efficient and customizable compared to many other gene editing tools. For instance, other gene targeting and editing tools like zinc finger and talons or transcription activator-like effector nucleases, require a complete redesign of the protein-DNA interface to change a target gene. Additionally, all the possible locations for cuts by the CRISPR-Cas9 complex can be easily kept in a genomic library of guide RNAs, making it really easy and simple to choose which genes to cut out. Thanks for watching. Please leave a like and subscribe.